All right, and I'm waiting for the thumbs up from Chris. We've got the thumbs up, so welcome our audiences that are joining from other counties. All right, we'll go ahead and get started again. We're going to have Dr. Lisa DeVetter talk to us about nutrient management. So please help me in welcoming Dr. Lisa DeVetter.
Great. Thank you, Boron, and good afternoon for our site locations. So I'm going to switch hats and put on a hat that's a lot more comfortable for me. I'm a trained horticulturalist and also sometimes studying soil science. And I'm going to be now talking as opposed to plant protection and now nutrient management and both blueberry and red raspberry. So the overall goal of this presentation is to not provide a comprehensive review. Really, that can take hours to provide a whole course to get you a good understanding of nutrient management, as well as get familiar with all of the wonderful research that has been going on in the Pacific Northwest on nutrient management. But the goal of my talk is to remind everybody of key concepts when it comes to nutrient management and to provide you some research updates that have come both from my research program as well as other research programs. And then also to point you towards available resources that are available should you have any questions about nutrient management in your crops. So when I think about nutrient management, I really begin thinking about what are the questions that we as growers need to be asking ourselves when we're developing a nutrient management strategy. The first one is, what are the requirements for nutrients? And again, I'm going to be using this pointer for our off-site locations. We need to know, based on the age of the planting, sometimes the cultivar of the plant as well, what the nutrient requirements are. And we're going to go over that in this talk here in a moment. We also want to know when are the nutrients needed, or when are the peak periods of demand, so that we can time application with those peak periods of demand. We also need to know how much of those nutrients are needed. That also varies on the type of plant, the type of cultivar, as well as the age of the plant and the amount of biomass that that plant is producing, both in vegetative structures as well as fruit that's harvested. I also want to know what's available in your soils for certain nutrients, particularly in a pre-plant situation, so you can target those nutrient amendments to make sure that when you're putting your plants in the ground, you're not running into a deficiency situation. And then it's also good to know what are the factors that are influencing uptake so that you can optimize those factors to ensure that your plants are more able to take up nutrients when it needs to at those peak periods of demand. And then lastly, you want to know about what are the sources of various nutrients depending on your production system. Are you organic? Are you conventional? Are you looking at foliar, granular, synthetic fertilizers? These are all questions to be thinking about as you develop your nutrient management plan. And then stepping back, it's always good to ask yourself as a grower, is my current nutrient management plan working? And I'm going to point to some specific tools that you can use to test your nutrient management plan and make some assessments to see if it needs to be modified. So let's go over on a review of nutrient management. So the goal of fertilization is to remove nutrient limitations before they become problematic or before nutrients become deficient. But on the other side too, we want to make sure that nutrients are not in excess, because we can run into growth problems if nutrients are excessive. So the traditional view of nutrient management is this barrel and stave concept, where these different staves represent requirements for crop growth and development. And by that I also mean by harvestable yield. On this side we have things that plants need for growth and development, light, water, but we also have our critical macro and micronutrients. And if any of these are limiting, we see yield start to decline. So we want to make sure that none of those factors are limiting. A nutrient is essential if it's required by a plant to complete its life cycle, and also from a grower perspective, when we're thinking about harvestable yield and making sure that we have a viable operation, we want that planting to be productive, not just that year, but in future years to come. So we're able to sustain our yields. Macronutrients are nutrients that are needed in large amounts by the plant, and micronutrients are nutrients that are needed in smaller amounts by the plants. So continuing on with the review, nutrients differ within their mobility within the plant, and that really depends on how the nutrients are moved and where they're moved in the plant body. So we have mobile nutrients, nitrogen, phosphorus, potassium, magnesium, and chloride. So these nutrients move in the phloem. And I think of these as the pipes in the plant that are connected all the way from the roots, all the way up to through the shoots and the leaves, and they move sugars in the plant. These can go both from the leaves down to the crown of the roots, which is our storage reservoir for carbohydrates and other nutrients. And they can also move from the roots and the crown up to the leaves during periods of need. And because they're able to be moved around and they're mobile, we see deficiency of these nutrients on older leaves. Because if there is a deficiency, the plant is able to move those nutrients to where they're stored and move them to a site where they're needed. And that's typically where newer growth is where they're going to be needed. Then we have immobile nutrients, or relatively immobile nutrients. It doesn't mean that they don't move. It means that relative to these mobile nutrients, they don't move as well. These nutrients include calcium, boron, sulfur, iron, manganese, copper, and zinc. These nutrients, they move in the xylem. And these are the pipes that move water in the plant. And they also move dissolved nutrient ions. And that's what these nutrients are, essentially. They're dissolved nutrient ions in water moving through the xylem. And these nutrients, because they're not mobile, we see deficiency on newer leaves or new areas of growth where we have nutrient demand. Just curious to get some interaction, because after lunch, I'm usually ready to take a nap. So how many of you have seen this slide before? It's really familiar, really common, but it's a great tool to think about the relationship between pH and nutrient availability. So what we have here are various macro and micronutrients represented in these different colored bars. And at the bottom here, we have pH. And here we're showing a pH of 4 all the way through 10. We rarely see beyond that in natural systems where we're having crop production. And if these nutrients are wider, it means that these nutrients are more available based on the soil pH. And if they're narrower, they're less available. And what I highlighted in red is the ideal pH for red raspberry. We are trying to target more of an acidic soil pH. 5.6 to 6.5 is ideal for red raspberry. And then blueberry, we're targeting an even more acidic pH range for them. We're targeting a pH of 4.5 to 5.5. If we go above or beyond these ideal ranges for these crops, which they're adapted to, we can see problems with deficiency and toxicity. So a good example for blueberry, if we have pH below 4.5, we can start having issues with aluminum toxicity. That's a very common symptom in low pH situations. If the pH is too high for blueberry, we have issues of iron deficiency, because the form of iron that blueberry takes up and utilizes in metabolism is not available at higher pHs. On top of that too, the preferred form of nitrogen for blueberry is not available, or is less available at these higher pHs than these lower pH ranges. So it's important that we aren't below these ranges or above these ranges for the particular crop. And then these two different plants that we're talking about, red raspberry and blueberry, they have different preferred forms of nitrogen. Again, this is really basic information, but I want to make sure that we're all on the same page, because I know in this audience we have growers of all types that might have varying levels of knowledge. So raspberry, adapted to slightly acidic to somewhat neutral soil pH, they prefer nitrate nitrogen. Whereas blueberry, adapted to lower soil pH, these are ammonium nitrogen feeders. So we want to remember that when we are providing nutrients, particularly nitrogen in this case. I think this is always good to go over, because we sometimes forget how plants take up nutrients. And this is important to know how we can optimize nutrient management and make sure that the plant is able to take up nutrients. So I feel like a lot of times we think a nutrient is just taken up by direct contact. The root's growing, it hits a nutrient, and bam, it takes it up. And that's interception and direct contact. That does happen, but it's really relatively minor form of nutrient uptake for a plant. Most nutrients are taken up by the system called mass flow. And mass flow is created by this transpiration stream. So this transpiration stream is created when we have plant leaves, which have these pores. Water exits from these pores. It's also a site of gas exchange. You know, a lot of you remember from high school biology, those pores take up carbon dioxide for photosynthesis, but they also lose water. And when the water is being lost from the leaves, a water tension is created. And what that does, it creates a tension in the xylem. And those are the pipes that are moving water. So the tension is in the leaves and the stems, and it goes all the way down to the roots. So you essentially kind of have like this straw or the stream of water moving up through the soil, into the roots, up through the plant body, and out of the leaves. And when you have dissolved nutrients in the soil water, they get taken up in this transpiration stream, and we refer to that as mass flow. And it's a main form of nutrient uptake in plants. And this is the most important for the nutrients nitrogen, calcium, and magnesium. And then lastly, we have diffusion. So going back, thinking about biology or chemistry, diffusion just simply means the movement of a solute or a chemical from high concentration to an area of low concentration in attempts to equilibrate. And nutrients do this as well. If they're a high concentration outside of the root, they have a tendency to move inside to the root where there's a lower concentration. And it typically continues to occur because as that nutrient is moved into the root, it's moved elsewhere in the plant to sites of growth and where it's being needed. And this is most important for phosphorus and potassium. So I mentioned, and I say this a lot, nutrients are dissolved in water and they're dissolved in their ion form. So when we're looking at nutrient uptake, we also want to make sure that we're providing sufficient water to our plants, because to take up a lot of these nutrients, there needs to be water for those nutrients to be dissolved in and for the plant to take them up. And I won't go over this in detail, but these are basically the ions that are nutrients, both positive cations and negative anions. So I want to go over again and talk about this transpiration stream just to kind of bring it back together again. We have water being lost from the leaves through evaporation. It creates this negative tension that pulls water from the roots all the way up through the plant body, and those dissolved nitrate ions are dissolved in that water and are moved in that system. And then we also have direct contact of a nutrient with the root. That's direct interception. It's a minor form of nutrient uptake. And then we have diffusion, where we just have a higher concentration of a nutrient outside of the root moving into the root where there's a lower concentration of that nutrient. And that continuously occurs because as that nutrient comes in, it's moved elsewhere in the plant. So we continuously have a lower concentration of that nutrient in the root. So let's talk about nutrient removal. So the reason we have to supply nutrients year after year after year in most situations is because when we're harvesting a crop, when we are pruning, when we remove that biomass, we're also removing nutrients that were stored in that biomass. So Bernadine Strict and one of our graduate students, Hannah Rampella, did some really good work with blueberry and raspberry in Oregon, where they looked at how much nutrients, specifically nitrogen, are removed in harvested fruit and in pruning woods. So they learned from blueberry, we're looking at 1.3 to 2.7 pounds of nitrogen being removed per ton of fruit. And then when we look at the prunings, they learned about 14 pounds of nitrogen are removed per acre of pruning wood. And I know sometimes we leave that pruning wood in the alleyways and it gets chopped up. Realistically, for blueberry, though, with such a small root system, it's very little of that nutrient that is in that pruning wood gets released and moved back into the raised bed system or the alleyways where those plants are growing. So those nutrients are removed. We need to look at replenishing them, and that's where a fertilizer application comes in. The same applies to raspberry, although we have different numbers. So per ton of fruit, we're looking at 3.5 pounds of nitrogen removed in the fruit. And then when it comes to pruning woods, we have about 17.3 pounds of nitrogen removed per acre of pruning. So let's look at the recommended nitrogen rates for raspberry. So the current recommendations, when we look at some previous research and are published in our Pacific Northwest Red Raspberry Handbook, which is currently in revision, is that when we have an established planting, we're looking at 50 to 80 pounds of nitrogen per acre. And then in the establishment years, we're looking at 30 to 50 pounds of nitrogen per acre. These recommendations, though, are based on granular applications when we're applying it the first half at primocane emergence and the second half right about a month before harvest. And we have these split applications so that we are optimizing the availability of nutrients during those peak periods of demands. We're working on developing recommendations for a fertigated situation, and I hope to see that in the newly revised version of the Northwest Red Raspberry Handbook. David Bryla, USDA scientist, horticulturist at the Ag Research Center, has been working on developing that, which again, hope to include in the newly revised Red Raspberry Handbook. For blueberry, we do have information in terms of what are the recommended amounts of nitrogen to be providing in a fertigated situation and in a situation where we're applying granular fertilizers. And we also have recommendations based on the year or the age of the planting in blueberry. And this is based on the work done by Bernadine Strick, my counterpart at Oregon State University, and Dave Bryla. A lot of this information is provided in their handbooks, which you can find on my website for free. When we're looking at granular applications, at the beginning of the years, when those root balls are really small, we're looking at hand applying or doing a higher rate if you're broadcast applying to take into account some inefficiencies with that broadcast application. Because again, when those blueberries are really young, they don't necessarily have a large root mass. So we can go with the lower rate of nitrogen with our granular application. If we're hand applying, you want to go on the higher rate for the granular application if you're broadcasting to take into account those inefficiencies. Also, when you're looking at granular applications, we're looking at splitting into thirds. So applying a third of that rate in April, the other third in May, and the final third in June. And that really coincides to when the blueberry plant is taking up nitrogen. So again, we're providing the nitrogen when the plant is taking it up and utilizing it. Another comment too on this table, if you're applying sawdust, which is really common in this area for blueberry systems, for every 3 inches of sawdust applied, there's going to be a mobilization of nitrogen because microorganisms in the soil, they're going to be utilizing that carbon, they're also going to be utilizing the nitrogen and essentially stealing that nitrogen from your blueberry plant. So to compensate for that, what you want to do is make sure that when you're applying sawdust mulch, you're applying about 25 pounds of additional nitrogen so that you're not starving your plants to do that microbial competition for nitrogen. Fertigation, sometimes it's questioned why for young plants we have higher rates of nitrogen recommended in a fertigated situation relative to a granular situation. This is really because to take into account the inefficiencies of fertigation when we have that small root mass again. So again, if you think of a new blueberry plant you just put in the ground, it's got a small root mass, and if you're applying nitrogen through drip, your spacings in the emitters, you're not going to be applying nitrogen all that nitrogen is not going to be getting to your blueberry root. So we tend to apply a higher rate to take into account nitrogen being applied where your roots are not located at. So another thing about nitrogen too, I think many of us know, is that more is not always better. There are issues that we can run into with regards to plant growth and development and how it impacts our yield if we apply too much nitrogen. And this is really, I'm focusing on blueberry here on this slide. One thing that we've seen in some of the research trials by Bernadine Strick is that if we apply too much nitrogen, we see reductions in fruit size and fruit firmness. So we can lose fruit quality, which can be really important if you're looking at a fresh market situation. And a lot of this is due to, we oftentimes see in other fruit crops, high nitrogen leads to softer fruit, but also if we're having a lot of vegetative growth, we can have a reduction in our berry size. Another issue is that if we are applying a lot of nitrogen to our blueberry plants, more than what the plant really needs, the plants remain growing into the late summer and the fall, and that can reduce our fruit bud set, which can have a negative impact on our crop the next year. Additionally, if we have nitrogen too much applied late in the season, we have the plants not being able to develop cold hardiness or acclimate, so we can see increased winter damage if we have too much nitrogen later in the season. Another thing too, and this is really particular for blueberries because they're very salt sensitive relative to red raspberries, if we apply too much nitrogen as well as other fertilizers, we can be causing salt damage unintentionally. And then on top of that too, if we're looking at higher rates of nitrogen, we can cause faster drops in pH, which might be good because we want lower pH in our blueberry crops, but we also want to make sure that we're not going too low with our soil pH, because then we can start running into issues of aluminum toxicity. So let's just take a quick look at the different types of nutrients and how they're formed. So we have dry, we have liquid, we have soil applied and foliar applied, we have organic and synthetic, we have controlled release. And then, what I want to spend some time focusing on is if the nutrient is organic, is it animal or plant-based? And I'll dig into that a little bit more in the next slide, because there's some really important considerations, particularly for blueberry, if we're looking at organic sources of nutrients, particularly with compost. So you should really consider the nutrient type when you're developing your nutrient management strategy, in large part because these organic sources have slow release, as opposed to these synthetic sources, they have faster release. And as I already kind of mentioned, we want to make sure that we're being careful with organic sources of nutrients. They can vary among batches, even from the same distributor. They can vary from batch to batch, and they can vary between years. So I really recommend that if you're using organic sources of nutrients, every single time you test it so that you have a good sense of what's being applied in your field. All right, so I said I kind of warn against using organic sources of nutrients, particularly compost and blueberry, because there can be some issues that we can run into, particularly some of the animal-based composts. And this is because they tend to be high in pH and high in salts, which can be a problem for blueberry because they are adapted to low pH conditions and they're not very tolerant to salt. So when we're looking at applying compost, our goal should be that material has a pH less than 6 and also an EC content less than 2 decisiemens per meter. So here's a reproduction of a slide that Dan Sullivan from Oregon State University and I put together, and we put in the Whatcom Egg Monthly. He went through really in his research program various types of animal source compost as well as some yard compost from leaf litter and mint. And he looked at the pH and he looked at the electrical conductivity. And so what I want to point out is that none of these materials were at or below a pH of 6, which should raise some flags if you're a blueberry grower looking at applying some of these materials. It's not really suitable with that regard. And then if we look at the EC content, again, our target is 2 for the most standard method of measuring EC. Nothing here except, the leaf was close to 2. So again, these should be raising a red flag that these materials could cause salt damage and also be negative in terms of impacting my soil pH. Now let's take a look at some of the more traditional organic materials that we add to blueberry, both peat and sawdust. If we look at their pH, they're 4.8, 5.2, and that's below 6. So that's the safe range we want to be looking at for our blueberry application. And they also have ECs below 2. All right, now let's shift gears and talk about tools and techniques to manage and monitor your nutrient sys program. So the first recommendation is to keep good records. So you can always go back if you have questions and you're looking at comparing information from one year to the next, you have the ability to do that. Soil testing is a valuable tool. We recommend doing that every 2 to 3 years unless there are problems. Tissue testing is a tool that I recommend you do every year to get a sense of nutrient levels in your plants, and you do it by cultivar. And then combined, when you have both soil testing and tissue testing as well as in-field observations, that can really help equip you to make assessments and judgments on your nutrient management programs and inform you on if you should be changing your practices to meet the needs of your crop. So let's just go over a little bit on soil sampling guidelines. The first thing I really recommend doing, particularly if it's a new lab, is to call the lab. Labs sometimes vary in their techniques and what they're looking for and how you should be preparing your sample. So just give them a quick call, let them know you want to submit a sample, and see what their recommendations are so that you're submitting a sample that is more likely to generate the results, the high quality results that you can interpret from. Sample consistently, so avoid areas of irregular soil types within your field situation. Sample those separately and avoid mixing so you have an understanding of soil type. in your area because they can lead to differences in nutrient management programs. We also recommend avoid spring sampling, particularly after fertilizing, because that can give you some false indications of what the actual nutrient content is in your soil. And then also in our situation where we're looking at rows, which we have in our raspberry and our blueberry crops, we want to make sure that we're sampling in the rows because that's really where a lot of our nutrients are applied banded. And we're also sampling within the zone of the drip emitters, where the roots are and where they're going to be experiencing your nutrient management program. Also, it's more important for blueberry, remove any mulch or other visible materials that might lead to misinterpretation in your soil sample report, and then also sample by representative blocks. And that kind of goes back to sampling consistently. And here, I'm not going to go over the full details. I'm happy to make these slides available. But here are the soil sufficiency levels for red raspberry. We don't have nitrogen on here because I don't recommend changing your nutrient management program based on soil nitrogen rates. Nitrogen is very dynamic in the soil, so what you see in the soil at one sampling point is not indicative of what you might see later on down the road. But we do have good ranges in terms of what sufficiency levels look like in the soil for phosphorus. Bray phosphorus is really something we tend to look at here in Western Washington with acidic soil conditions. If you have more alkaline soil conditions, you're going to be focusing on the Olsen test. And then we have recommendations for potassium, calcium, manganese, and boron. And again, make sure your pH is within that target range. And then this is the same information except for blueberry. Again, focus on your pH, pay attention to your EC because high salt content can be a problem for blueberry. They are sensitive. And again, these are the recommended ranges for the various nutrients and soils. Now let's shift gears and talk about tissue sampling. And this is a really useful tool to being able to assess how your nutrient management program fared that year. It's not really going to tell you what I can do right now at this particular point in time to correct any problems, but it can tell you what I need to do differently for next year and years thereafter. So the first thing is to make sure that you're collecting the right tissue. And that includes the most recent fully expanded leaves. For blueberry, the traditional recommendation is below the fruiting cluster and not a whip. The whips are those vigorously growing tissues coming from the crown or the roots or elsewhere lower on the plant. And then for raspberry, we're looking at targeting primicanes about 12 inches from the tip or the apex. You also want to sample problematic, if you have problematic plants and you're trying to discern if it's a nutrient management problem or something else, you can sample the problematic plants and keep that sample separate from the asymptomatic plants. And that can tell you if your problem is a nutrient situation or maybe something else like a soil-borne disease problem. And also we recommend to sample cultivars separately. Also, we're typically looking at sampling late July to early August. That's when nutrient levels kind of stabilize and plateau. And that's a good time where we can compare the levels that we see in our field to establish standards. As I said with soil sampling, I think it's always good, especially a new lab, to call the lab, let them know that you want to send some samples so that they're prepared and they can give you any guidelines in terms of how they want to receive that sample to ensure quality results. Generally, I don't recommend washing leaves or soaking them because that can leach the potassium and lead to incorrect results on your leaf potassium levels. And then also, this is where note-taking can come into play. Note that if you've applied any fungicides or foliar nutrients, and even dust, because those can influence your results. And then just like I mentioned earlier, when you're looking at and interpreting these results, it's always good to pair them with observations in the field and having good records. These are the recommended sufficiency levels of tissue nutrients in red raspberry, and these really apply to that late July, early August sampling period. Again, I'm not going to go through these bit by bit, but I'm happy to share the slide. I point you to resources so that later on when you're interpreting your tissue analysis results, you have this information available. And then for blueberry, I think this is also important to know that there have been revised standards for northern highbush blueberry for various nutrients. And a lot of this has been coming from Dr. Bernadine Strick's research program. And we've revised the nutrient standards based on some previous experiments and analyses of leaf tissue contents and harvestable yield. Again, I'm happy to share these for you. So now I want to shift gears. I did a quick update just to make sure we're on the same page and provide some good tools and information for thinking about nutrients and monitoring your nutrient management program. So now I want to talk about some research updates for various projects that have begun or initiated in my program. So the first one is the impact of canopy and leaf position on tissue sampling. I see a couple of cooperators in the audience who, Joan Davenport, who's a soil scientist, and Prosser and I have worked with. Essentially what we did in this project, which is still ongoing, is we sampled leaves in Western Washington and in Eastern Washington. And for this particular project, we chose to focus on Draper. And what we did is we intensively sampled in late July at different canopy positions in the plant, high, medium, and low. And then within those 3 canopy positions, we sampled at different branch positions. High, so at the upper position, middle, which is the shoot immediately below the cluster. That's traditionally what's recommended for leaf tissue sampling. And then at the very bottom. And we finally have results from that. I think, I've really been eager to see, because Joan has been looking at extracting some of the nutrients and looking at some of the data. Here are some of the data from this. It's been really interesting. So what we saw was that leaf tissue nutrients varied by location, and I'll dig into that a little bit more on the next slide, but they didn't differ by leaf position. So it didn't matter if we sampled on that branch high, beneath the cluster, or low. What did matter for 8 nutrients, these nutrients here, nitrogen, phosphorus, potassium, manganese, sulfur, copper, manganese, and zinc, that we saw differences in nutrient concentration that were statistically significant based on position in the canopy. So if it was high, middle, or lower in the canopy. And that's what this figure tries to show. So I mentioned we did this in Central Washington in the Prosser area, and we did this in Western Washington. What we see here is on the y-axis, percent nitrogen in our leaf sample. And then we have lower, middle, and upper portions of the canopy for Central Washington. And we have lower, middle, and upper portions of the canopy in Western Washington. And consistently, we saw higher concentration of nitrogen at that lower position in leaf canopy than we did in the upper position. So we're going to be continuing on with this experiment in the subsequent year, so 2017. And from this, we might be recommending different strategies for leaf tissue sampling. So on this trial, we also are looking at other cultivars beyond Draper. We're sampling leaves for Duke, Draper, and Liberty in Western Washington, and Aurora for Eastern Washington. And the reason we selected those cultivars is because they have different fruiting periods, Duke being early, Draper being mid-season, and Liberty and Aurora being the late season. And we did this to see how tissue nutrient concentrations varied within these cultivars and really between these locations. Western Washington has a maritime climate, and Central Washington, where we have high pH soils and a lot warmer growing conditions. And what did we learn? So this is still ongoing work, but what this slide shows is percent nitrogen concentration for Eastern Washington Duke, which is in this blue line, Western Washington Duke in this orange line, Eastern Washington Draper, which is gray, Western Washington Draper, which is kind of this orange color, and then Aurora, which was just in Eastern Washington, and Liberty, which was in Western Washington. And so overall, what we see, nitrogen concentration was lower in Eastern Washington relative to Western Washington, and we didn't see a lot of differences in cultivars, which corroborates some of the work that Dr. Bernice Strick has been doing in her program. So where do these levels lie with regards to known standards? So what I'm highlighting here, these are the Michigan State standards for nitrogen, and these are the Oregon State University standards that Dr. Bernice Strick has helped develop. So if we look at our periods where we're sampling nutrients, we're slightly below for nitrogen in Western Washington. And then for Eastern Washington, we are below those recommended guidelines. Yet these are productive plantings that we're sampling from. So again, this project is really helping us try to think, well, what are the sufficiency standards for these cultivars under these different growing conditions. Now I want to highlight some additional work that's been going on in my program where we looked specifically at boron and calcium in blueberry. This is work that's really being led by my graduate student, Matt Arrington, as part of his PhD program. And we're looking at the impact that boron and calcium has on improving fruit set and yield, as well as fruit quality. And that's more of an interest for calcium. So why did we choose these nutrients? We chose boron because it's a critical micronutrient for plant growth and development. It has limited mobility within the plants, particularly blueberry, and we often struggle with having adequate amounts of boron in blueberry here in Western Washington. Another thing that we struggle with Western Washington blueberry is we have low fruit set. This could be due to pollination. It could also be due to boron. So we're trying to separate that out and see how much is it due to boron, how much is it due to pollination. But we do know from other research that boron does improve pollen health. And it also improves other reproductive processes that can help improve fruit set and yield. So that's why we're interested in boron. Why we are interested in calcium? Well, calcium has many important physiological roles and functions. A lot of it is pertaining to fruit quality. We really started focusing in on calcium for Draper though, because we have a condition known as premature fruit drop in Draper, particularly in Northwest Washington and Whatcom County, as well as in British Columbia. And it can be significant under certain years. And some previous work done by Erica Brandt in British Columbia has found that foliar applications of calcium can reduce premature fruit drop. So we're looking at it, supporting some of that research and seeing what role calcium has in reducing premature fruit drop and how it interacts with boron. So we had several experiments that Matt has conducted over the years. So the first experiment, we just focused on boron, and we had these 5 treatments applied in Duke plants. Control of no boron, spring applied boron at a low rate at 8 ounces to the acre, another spring applied treatment at a high rate. Again, these are all within label. And then we looked at fall applications, both in a low and a high rate. And we utilized this product, Nui Boron 10%, which was recommended by the co-op or CHS at the time as a product that growers are utilizing. And it really wasn't, maybe the most exciting study because we didn't see a lot of effects due to our foliar boron applications. We didn't see high amounts of fruit set due to our treatments of boron. What we did see more than anything was a year effect. In 2015, we saw lower fruit set than in 2016, and we attribute that to climactic conditions. So again, we didn't see any effect due to foliar applied boron on increasing fruit set, estimated yield, and berry size. However, when we look at our leaf tissue nutrient concentrations, we did see that concentrations were slightly, but not significantly higher with our fall applications. And then there was a second experiment that we did, and this is an experiment to look at the interaction of boron and calcium, and we did this trial with both blue crop and Draper. So this table's maybe a little bit fuzzy, but we kind of modified our treatments as the years progressed, and we learned more about how these treatments were faring. So in both years, we had untreated plants where we didn't apply any foliar boron or calcium. In 2015, we had a low rate of boron. We used Solubar for this experiment, and we had a high rate in 2015 and 2016. We dropped the low treatment or the low concentration because we didn't see any treatment effects in 2015. So we wanted to drop that so we can substitute something else to look at that might have more effects. In 2015, we also looked at a low rate of calcium using a product called Phytocal, which is calcium chloride. We use this product because we were working with some organic cooperators for this research, and that is an organic approved material. We also looked at this material at a high rate in both 2015 and 2016. And then we had some alternative products. In 2016, we looked at a product called Liberate. And then we also looked at boron with calcium, both at a low rate and a high rate. These are still part of the PHYTA program that are approved for organic situations. And then in 2016, we also looked at a boron plus manganese. And we looked at that because there were some previous results that made this product look encouraging. So what did we learn? We did see, which was interesting, decreasing fruit drop in our Draper treatments treated with calcium, particularly at some of the higher rates. And this corroborates some of the work that Erica Brandt has done, where we've seen Foliar applied calcium, reducing premature fruit drop in Draper. And what this plot shows is berries drop per plot, and we have our various treatments over time. Really what these bars represent are different sampling points within a period that Matt went out and looked at the amount of fruit drop on the ground. And the take-home message is that anytime we have calcium in the mixture, we are seeing a reduction in fruit drop. What Eric has seen at higher rates is that he's seeing even less fruit drop with higher rates of calcium. When we dig into that data a little bit more, we didn't see any differences in pollen growth rates. So Matt applied the treatments and then he collected pollen and he germinated them on media plates and looked at the rate of pollen germination and the rate of pollen tube growth. And here's a picture of some of what the treatments or samples look like, and he didn't see any difference based on treatment. So, you know, based on this experiment, we didn't see any impact on pollen growth rates due to boron treatments. And we also saw no difference due to fruit size and fruit firmness based on any of the boron or calcium treatments that we looked at. We also didn't see any differences in our estimated yield. So really, we didn't see any treatment effect except the reduction in premature fruit drop in Draper. So just some general conclusions from those trials. I also want to mention we didn't apply any surfactants for these particular treatments. We applied them according to the label, and the label did not recommend surfactants. So that's why we did not use surfactants. We saw a few consistent results in some of these trials. Particularly the first experiment, we really saw a year having the largest effect, few treatment effects. Foliar applications of boron under our conditions of the experiment did not increase fruit set estimated yields or any attributes of berry quality that we looked at. Foliar applied calcium did reduce fruit drop in Draper in 2016 only. And it was only 2016 and 2015, if anyone remembers, that was a low Draper drop yield, which is interesting and provides another opportunity for us to kind of dig in and look a little bit more in terms of why Draper drops fruit under certain environmental conditions. And that's going to be some future work that I'll talk about in just a moment. And again, I want to make sure that we're recognizing Eric Brandt and that how these results really corroborate some of what he's finding in Canada, where foliar applications do seem to reduce premature fruit drop in Draper. And so I asked Eric if he would be willing to share a few slides just to demonstrate what he's seen up in British Columbia with his treatments. We have fruit drop here shown on the y-axis and percentage, and then we have concentration of fruit calcium. The main take-home message is that the more fruit calcium we have, the less fruit drop we are seeing. And this is 2016. Also just anything, and if you're wondering what this picture here is on the side, what's pretty characteristic in Draper when it drops the fruit is that if you take a cross section and look inside, we see this brown tissue, and that's indicative of a calcium deficiency, and that's why we, the very beginning, started looking at calcium. And then here, just want to highlight that the relationship between fruit calcium and fruit drop is quite high and significant. So future research, we're really curious, both Eric and myself, to kind of dig in and try to understand more about the physiology and what's causing premature fruit drop, because I don't think it's just calcium alone. So let's kind of take a step back and think about how calcium moves in the plants. Go back and think about the transpiration stream. So we have calcium moving in the xylem, those water pipes. We have that negative tension being created from water being moved from the leaves. And we tend to see preferential movement, particularly for Draper, among all of our cultivars, for calcium to be moved at higher concentrations into leaf tissue than to fruit tissue. And that's what I'm trying to show in this little picture here, where we have more calcium in the leaves relative to the fruits, which kind of creates this deficiency situation. We have less calcium in the fruit because of preferential partitioning of calcium in the leaves. One other observation that we've seen with Draper drop is that it seems more severe under high vigor situations. I think that's because we have more leaf tissue, more calcium moving to those leaves, and less calcium moving to the fruit, further exacerbating this deficiency situation. The other thing too that kind of adds a piece to this puzzle that we're going to be looking at a little bit more in 2017 is that fruit drop seems worse under years where we have reduced solar radiation or high cloud cover. And the reason I think that is because when there's clouds, those holes, those stomata close up, and those are really important for causing calcium to move in the plant. And I think that aggravates the deficiency situation. So Eric and I, as well as a few others, were looking at how physiology and environmental conditions like solar radiation influence fruit drop, and we've been collecting samples over the years from British Columbia, Washington, all the way down to Oregon. And it looks like a pretty strong relationship in that high cloud cover leads to more fruit drop, high vigor leads to more fruit drop, and that calcium can mitigate that. What Eric is going to add, or excuse me, what Matt is going to add to this puzzle is that we're going to do some shade experiments in Draper fields in 2017 and induce this cloud cover situation or kind of mimic it and apply calcium. And so we're hoping to see if we apply shade, does it lead to more fruit drop? And if we apply calcium under those shade covers, does it impact and reduce fruit drop? And that'll just kind of help us get a better understanding of the role calcium and cloud cover has, or these other environmental conditions have in fruit drop. All right, as I wrap up, as I think I'm cutting close on my time, I want to point you to these guidelines. The nutrient management guideline for blueberry is currently in revision, but it's still a great resource to look for within the PNW extension handbooks. We also have one for caneberries. Both of these are available for free on my website. So if you haven't visited my website in the past, here's the URL. And I try to put a lot of resources available up there to help growers if they're looking for questions and answers. I'm also available by email or by phone. And with that, I want to thank you all for your attention and see if we have time for a few questions. We have one question. Yes.
Is the nitrogen migration specific to Draper?
Sure.
Is it just Draper or preferential to leaves or is it another?
Sure. So the question was the kind of the distribution of calcium to fruit and leaves and it being greater in the leaves. Is that just for Draper? No, it's not just for Draper. However, Draper is more prone to allocating more calcium to the leaves than fruit. We see it in other cultivars, but that difference is just much greater in Draper, particularly under high vigor situations. So, all right, thank you.
Let's give Lisa a big round of applause.